In our previous segment, we have discussed about the vectors that are vehicles of recombinant DNA technology. We have seen the various properties of vector that any molecule must possess in order to behave or act as an ideal cloning vectors. Right? So, in this segment, we will start with the various vectors that are present within the host cells. Say, for example, various prokaryotic cells, eukaryotic cells, plant-based vectors are also available for experimental purpose as well as for research purpose. So we will be starting with Pdr3 vector. This is the first artificial cloning vector, right? It was discovered first artificial cloning vector. It was discovered in year 1977, and the name of the vector is Pdr3 Vector. Let us see or understand how the naming it is done. Pdr3 Vector. P stands for plasmid. Right? P stands for plasmid. B stands for Bolivar. Boliver. R stands for Rodriguez. Rodriguez. So P stands for plasmid, B stands for Bolivar, R stands for Rodriguez. The name of the scientist who has discovered Pdr3 right? So Br is the name of the scientist, P stands for plasmid, and 322 indicate the type of strain, right? 322 is the code or the strain number that was first discovered. So Pdr3 is an artificial cloning vector discovered in the year 1977 by Bolivar and Rodriguez. P stands for plasmid, Br stands for Bolivar and Rodriguez. 322 is basically the strain number or the code number, whatever you want to say. Right? So this plasmid is basically a typical plasmid that has all the properties that we have discussed in the previous segment. Right? This Pdr3 has one origin of replication site. Apart from this, it also has one ROP site, that is protein of replication. Other than that, this Pdr3 has two selectable markers which are antibiotic resistant gene only. They are antibiotic resistant gene. And total 8 cloning sites, 8 restriction or cloning site this Pdr3 has. Now we will draw the same diagram that that is given in the NCERT, and we will understand the locations of different restriction endonuclease enzymes, location of ROP, ORI, as well as antibiotic resistant gene. Right? So plasmids Pdr3. Before discussing into the details of all these things, basically there are two types of molecules or the vectors which are used in recombinant DNA technology. The first one is plasmid and the second is bacteriophage. Plasmid is basically extra chromosomal DNA which works or functions on its own. So it is a kind of autonomous structure. So it is autonomous circular extra chromosomal DNA within the bacterial cell. It can work on its own because it has all these structures. Bacteriophages are viruses that infects bacteria. Right? So bacteriophages are the viruses that infect bacteria. Then we have to focus on one more important term, that is copy number. Copy number of plasmid is very important when we are dealing with this type of experiments. Copy number means the number of the copies that a particular plasmid has in a particular cell. So copy number is number of copies of a plasmid what we call it as copy number. Normally it has one copy number per cell. It can reach from 1 to 100 per cell can also be the copy number of a given classmate at a given point of time. Right? Now let us come back to Pdr3 So Pdr3 is autonomous circular extra chromosomal DNA. So we will draw the diagram that is being drawn in the NCERT because same diagram we have to follow for exams also. Okay. Here is the site where two restriction endonucleases are present, that is CLA1 as well as HIND3. This is a site for CLA1 and here HIND3. If you remember, HIND2 was the first restriction endonuclease discovered, from which bacterium, that is Haemophilus influenzae. HIND3 is also isolated from Haemophilus influenzae. After drawing and understanding all these restriction sites, ROP site, ORI site and antibiotic resistant gene, we will also write from which from which bacterium cells all these 8 restriction endonuclease enzymes has been isolated. So CLA1, HIND3, and there is EcoR1 site also. EcoR1 site. Then here BAM, H1 and SAL1 are located. All these are restriction endonucleases. BAM, H1 and here SAL1. Right? Then within this segment we have PVU2 restriction endonuclease. Right? Then afterwards we have PVU. Okay. Then here we have origin of replication, that also we will write. Apart from this, within this segment we have PST1 restriction endonuclease site, and here we have PVU1 restriction endonuclease site. Right? So starting from CLA1, then we have HIND3, afterwards we have BAM H1, SAL1, right? Then this is a vacant place, we have PVU2 restriction endonuclease, then this is again vacant. Afterwards we have ORI site. Let me use ORI site here. After ORI we have PST1 restriction endonuclease, then PVU1. This is PVU2 and PVU1, isolated from the same bacterium, but one is isolated first, 2 is isolated the second time. Here in the region of PVU2 we have ROP protein. So let us write ROP protein, and near here we have tetracycline resistant gene which is present, and within this segment we have ampicillin resistant gene also present. Right? So this Pdr3 acts as a typical or ideal cloning vector, most extensively widely used cloning vector in the recombinant DNA technology. Ampicillin resistant, tetracycline resistant. They are antibiotic resistant genes which are acting as a selectable marker, which will select the recombinant ones or the recombinant cell from the non-recombinant cell. Okay. ORI segment, ORI region, one ORI is present. One ROP, that is protein of replication present, which will initiate the replication process. Right? 8 restriction or the cloning sites. Let us count 1, 2, 3, 4, 5, 6, 7 as well as 8 restriction or cloning sites are present. Okay. Now let us write these restriction endonucleases: BAM H1, CLA1, HIND3, EcoR1, PST1, PVU1, PVU2, like that. From which bacterium they have been isolated. So it can be asked as a 4-mark MCQ in the NEET, although it is not given in NCERT. So first of all, we will write for hint 3. Hint 3 we know it is from Haemophilus influenzae. So hint 3 is isolated from Haemophilus influenzae. Then BAM H1 is isolated from Bacillus amyloidoic. So Bacillus amyloidoic. SARS-1 is isolated from Streptomyces albus. Streptomyces albus. PVU2 is isolated from Proteus vulgaris. Proteus vulgaris. Then PST1 is isolated from Provindicia storti. So it is isolated from Provindicia storti. PROVINDINCIA STUARTI. All these are the names of the bacterium from which they have been isolated. PVU1, PVU2. Proteus vulgaris, the same bacterium. But it is isolated first and to indicate it is isolated second time. Or the second. EcoR1 from the normal bacteria, that is the favorite one. Escherichia coli, that is E. coli. And the remaining one is the last, that is CLAV1. Cryophenon latum. Cryophenon latum is the bacterium from which it has been isolated. Right? Then afterwards, the different plasmids are available, which are artificially created in the lab, or they are naturally available also. The second category of the plasmid is PUC plasmid. PUC family plasmids. Right? P stands for plasmid. UC, that is University of California. They were discovered in the University of California, and there are various plasmids that fall under the category of PUC. That's why it is called as PUC family plasmids. And we will discuss the most common one, that is PUC8. So PUC, P stands for plasmid, U and C stands for University of California, which have isolated these PUC family plasmids. And in the PUC family plasmid, the most common one that we are going to discuss is PUC8. And one point we will discuss, that is the selectable marker. Marker is a substance or a gene which will mark the transformed cell or the non-transformed cell. So here the selectable marker is LAGZ gene, which we know it is an enzyme producing gene. Right? Antibiotic resistance is not present. Here AMP and DET, ampicillin and tetracycline, are the two genes which are acting as a selectable marker. In case of PUC8 or the PUC family vectors, the enzyme producing gene is acting as a selectable marker. And which enzyme it is? Beta galactosylases. Right? Then the third viruses or the third category of plasmids within the bacteria, as well as eukaryotic organisms, also can act as a plasmid, or they must have the plasmid yeast. It is BAC and YAC. BAC, that is bacterial artificial chromosomes, which are acting as a plasmid. BAC, bacterial artificial chromosome. Then within the yeast family, there is one plasmid which is called as yeast artificial chromosome. And we all know this YAC and LAC were discovered during HGP Human Genome Project, but YAC was used in extensively used in human genome project. Right? Apart from this, we can have bacteriophages also acting as a plasmid. What are bacteriophages? They are the viruses that infect the bacteria. So bacteriophages include the lambda-phage, M13-phage and retroviruses. Bacteriophages includes lambda-phage. Examples are very important. Second is M13-phage. Then in the category of the viruses, we have adeno, retro and papillomaviruses can also act as a plasmid because they have the capacity to transform normal cell into the cancerous cell. So we have retro, adeno and papillomaviruses also acting as the plasmids under the category of bacteriophages. We can also have the plant-based vectors also, that is Ti plasmid as well as Ri plasmid. So plant-based vectors. The first one is Ti plasmid, that is the most common one which is used. Ti plasmid, that is P stands for tumor, I stands for inducing. This plasmid induces tumor into the normal cells. That means normal cells are changed or converted or transformed into the tumor or the cancer cells. So Ti plasmid is tumor inducing plasmid that is isolated from bacterium, agrobacterium tumor facies. So isolated from agrobacterium tumor facies, and it is also called as tumor inducing plasmid. It transfers a piece of DNA which is called as tDNA. That is basically tumor inducing DNA that converts or transform normal cells into tumor cells, and a mass of cells get accumulated on the region of the plant, and then it is basically called as crown gall disease. Right? What are the basic advantages of using Ti plasmid in the recombinant technology? And one thing we need to remember for this Ti plasmid, that it can be used in plant cells only. We cannot use Ti plasmid in any other cells except plants. Right? The advantage of tDNA, that is their transfer DNA or human DNA, is it goes directly to the host cell genome and starts replication there. And the second is it also initiates the cell division. So it stimulates the cell division. Then it goes directly to the host DNA. And starts or initiates replication. Right? Then the second category within our second example, that is Ri plasmid, root inducing plasmids from Agrobacterium rhizobium. So Ri plasmid, that is root inducing plasmid. And this root inducing plasmid is isolated from Agrobacterium rhizobium. Rhizobium, that is a genus, Agrobacterium rhizobium. So this is all about the vectors, the various vectors which are used in the prokaryotic cell. Eukaryotic cell includes the viruses, the yeast eukaryotic cell is there, then plant-based vectors. In the next segment, we will discuss the techniques or how to make the host cell competent to take this DNA of